The Gram reaction is usually the first test for diagnosis of a pathogen. It distinguishes between gram-positive and gram-negative bacteria. Gram-positive bacteria have a thick peptidoglycan wall and are stained purple, while gram-negative bacteria have a thinner peptidoglycan wall and an outer membrane and are stained pink. Begin by lighting your Bunsen burner to keep an updraft of bacteria away from your sample. Next, heat and cool your loop. Since we are using a sample from an agar plate, we must add 10 to 20 microliters of water with a pipette or loop and place it on the glass slide. Once completed, heat and cool your loop again. Then lightly touch the surface of a single isolated colony off the agar plate and mix with a drop of water. Alternatively, if we had a fresh liquid culture, we would make a thin layer of bacteria cells on a clean glass slide. Allow the bacterial suspension to air dry completely. There should be no visible opalescence. If you see a white blob, it's too thick. Once you have finished placing the sample on the slide, heat your loop once again and allow your slide to air dry. Once dry, hold the slide with your clothespin and fix the specimen to the glass slide by moving the slide 3 times through the Bunsen burner flame. If you forget to fix your cells, they will wash off during the rest of the procedure. After heat fixing, place the slide on the staining rack. Then gently flood the smear with crystal violet, your primary stain. After 1 minute, tilt the slide and let the crystal violet run off. Next, gently flood the smear with Grimm's iodine, the mordant. The addition of iodine creates a complex with the crystal violet, making it difficult to lead the peptidoglycan wall of gram-positive bacteria. After 1 minute, rinse the slide briefly with water. Using an indirect stream is best to prevent the loss of any cells from the pressure of the water. Next, hold the slide with the clothespin at an angle and add 95% ethanol drop by drop to the slide until the ethanol runs clear and no color comes off the slide. This is about 10 to 30 seconds. Ethanol acts as the decolorizer. Then, rinse the smear with water for 2 seconds. Next, gently flood the smear with saccharin, the counterstain. After 30 to 60 seconds, rinse the smear with water for 2 seconds. Let the slide air dry and blot with bibulous paper. This is what the slide should look like after blotting. Once dry, add a drop of immersion oil onto your smear and examine the slide with the microscope under the 100x lens. You will not be able to see and distinguish bacteria with a 10x lens. Here is an example of a mixed culture. The gram-positive bacteria appear violet, while the gram-negative bacteria appear pinkish red. Note that human cheek cells do not show a gram reaction as they do not have a bacterial cell wall. Often it can be difficult to distinguish between gram-positive and gram-negative organisms, but don't lose hope. Microbiology is not always an exact science, but with practice, it will become easier to distinguish between these 2 types of bacteria.